Okay, so we need to explain. So first of all, what we're doing is we're using an optical microscope. Straight away, if we're using an optical microscope, we know that this is using a wavelength of light to magnify. Okay, and we're given that the piece of leaf tissue observed was very thin. Okay, explain why this is important. Now, this is literally a model flashcard answer. So make sure that you make this in your notes. Make sure you note it down because it's a very common exam question. I've picked out another exam question which is literally from a different year, but the same question, different context, but the same mock scheme answer. So why is this important? It's a single layer, okay? It's a very thin layer. And the reason why it's important is so that you can form a single layer of cells. So to form a single layer of cells, so the light can pass through. And that's all you have to say. Okay, so very common exam question. This is the mock scheme. It says single few layer of cells so light can pass through. Okay, imagine the specimen, imagine the piece of leaf tissue that we obtained was very thick. If it was very thick, then the light wouldn't have been even able to pass through. If it doesn't pass through, then we can't see and observe the leaf tissue in detail. Okay, we won't even be able to see all of it. So it's really important that the specimen that we view is very thin. So it avoids the layers of cells from rolling over each other. We want to see a clear layer of cells and a single layer of cells. We don't want overlapping layers of cells. We want just a single layer of cells. So just a single layer like that, a single layer of cells, so we can view it all the way through and see the structure in more detail. We don't want a structure like this. Let's say that's a cell. We don't want a layer like this. Okay, we don't want something like this. Okay, so as you can see, this is a single layer of cells. And the reason why we got this is because we have a thin leaf tissue. That thin leaf tissue gives us this single layer of cells. Over here, we have multiple layers of cells. So we have multiple layers of cells. Because we have multiple layers of cells, what does that mean? We can't view the cells clearly. This means that the specimen is thick. So the leaf tissue that we've obtained is thick and it's not allowing all the light to pass through. Okay, there are multiple layers of cell, which means that this is unclear. Okay, we can't view it in detail. Whereas over here, we have a single layer of cells. Because we have that single layer of cells, we can view this in detail. Okay, so that's the exam question. Very common exam question. Make sure that you make a note of this mock scheme. This is a very common mock scheme answer. Okay, moving on to another very common exam question. It says the student produced a biological drawing of the leaf tissue they viewed through the optical microscope. Give 3 ways the student could ensure they produce a correct biological drawing of the leaf tissue. Assume that the student uses a sharp pencil. Okay, so this is more about, there's a required practical on this, on microscopy that you will do. This is more related to the required practical, but it always comes up. And the only way that you can answer this is by looking at mock schemes. Okay, the more you look at mock schemes, the more you will know to answer questions like these. So how do we know that we have produced a biological drawing? Okay, so first thing, any time a question like this comes, write down, make sure you show a magnification scale. Okay, because for a biological drawing, you will always need to have a magnification scale. Okay, so the first go-to one is the point about magnification. The magnification scale needs to be shown. Secondly, in a biological drawing, you need to know what is what, because nobody can read each other's drawings unless it's like really good. Um, so you need to show labels. Okay, so you need to add labels to the drawing. Okay, and then thirdly, sorry, um, you basically have to say what you shouldn't do. When you're drawing a biological drawing, there should be no sketching. There should be no sketching or shading. Okay, so um, you need to make sure that or there is no shading or sketching. So those are the three go-to ones that I would always write down. Um, Literally going down, that's my priority order. Okay, always remember that all biological um, drawings need to have a magnification scales. It is often missed on many people's biological drawings. You need to add labels to the drawings, and you need to make sure that it's a continuous line, and there is no shading or coloring or sketching or anything like that. Literally one continuous line, and that's what you call a biological drawing. Here's the mock scheme. So it says use single continuous joined lines. Do not use sketching, no crossing or hanging lines. Do not use shading or hatching. Draw the outline of tissues only. Do not draw the cells in detail. Draw parts to the same scale or relative size. Make sure you show the labels and then you show the magnification. Okay, so all of those points there are really important, but the three that I have listed are the easiest ones to remember, and they are the most common ones that you would use in an exam. Okay. Moving on. So as I said before, the first question that we looked at, it is a very common exam question. Over here it says a very similar, if not the same sort of question. A scientist prepared alveolar tissue to view using an optical microscope. The scientist cut very thin slices of the alveolar tissue. Explain why the scientist used very thin slices of alveolar tissue with the optical microscope. Over here, explain why this is important to use leaf tissue, which was very thin. And over here, it says, explain why it is important that we use thin slices of alveolar tissue. So it's going to be the same mock scheme answer. It creates a single layer of cells so light can pass through. Exact same answer. This question was taken from a different year's paper. That first question was from a different year's paper. However, both of them have the same mock scheme answer, which emphasizes the fact that you need to learn these mock scheme points. You know, for common exam questions, this is definitely one of them. Learn this mock scheme answer, and if it comes up again, you will definitely know what to write. You won't be spending 2 minutes on a question like this. It will literally be 30 seconds, move on. Okay. Next question is a calculation question. It says the scientist used a ruler to measure the diameter of some of the alveoli. The table below shows the scientist's results. Okay, and we're given the diameter of the alveoli. in millimeters. The magnification of the image in the figure above is times 40. Use this information and the table above to calculate the mean diameter um, in micrometers of the alveoli. So it says in micrometers. So the first thing that we're going to do is it says the mean diameter. So what we're going to do, let's work out the mean first. Okay, so to work out the mean, all you would do is you would do 4, add 2, add 5, add 1, add 2, add 3, add 5, add 2, and then you would divide that by the number of times we measured the diameter. So 1, 2, 3, 4, 5, 6, 7, 8. So we've measured it 8 times, so we're going to divide it by 8 because the mean is when you add them all together and you divide by how many times you've calculated the mean. So if I type that into my calculator, so it's going to be 4 plus 2 plus 5 plus 1 plus 2 plus 3 plus 5 plus 2. That's going to give me 24. And then we're going to divide it by 8, and that's going to give me 3. So the mean diameter is 3 millimeters. Now, because the mean diameter is 3 millimeters, and we've underlined a key bit of information over here, it says micrometers. So in terms of micrometers, how do we get from 3 millimeters to micrometers? We need to multiply by 1,000. So if we multiply that by 1,000, we're going to get 3,000 micrometers. Okay, so we've got 3,000 micrometers, and this is our image size. Yeah, used a ruler to measure the diameter. So this over here is our image size. Okay, we know our magnification is times 40. So what we're going to do is so I equals A multiplied by M. Image size is 3,000. And we need to work out the actual size, and the magnification is 40. So all we're going to do is going to be 3,000 divided by 40. And if we type that into our calculator, that's going to give us an answer of 75 micrometers. Okay, so 75 micrometers for 2 marks. Okay, and there's our working out. 2 marks for 75, 1 mark for evidence of writing out this formula, or the word equation, correct denominator, which means that you divide by 40, or correct numerator, what you're dividing. Okay, anything like that will get you 1 mark, 2 marks for the correct answer. Okay. The next question, it says the figure below shows a transmission electron microscope image of the Streptococcus bacteria. Describe how the appearance of the area containing DNA in a TEM image of a eukaryotic cell would differ from that shown in the figure. Okay, so this is not really an electron microscope question. This is the image of the bacteria. Okay, and we are asked how will the image differ if we had a eukaryotic cell instead of that bacteria. Okay, what does a eukaryotic cell have that a prokaryotic cell doesn't have? A eukaryotic cell has a nucleus. So what you would see is that you would see a nucleus, and there's 2 marks, so we're going to have to list 2. So you would have to say nucleus. What else will we not see? We also won't see. Let's have a think about it. So if you have a look at this image, we would also not see. So it says components. What you would not see. So we wouldn't see a nucleus. In a prokaryotic cell. In a eukaryotic cell, we would see a nucleus. We would also see. So this is a bacterial cell. We would also see mitochondria. Okay, and the reason this will be mitochondria, which contains DNA. Okay, I've seen multiple questions like this, and they normally catch people out. what you're spotting for the difference is basically the DNA. Okay, so first of all, the nucleus is going to be present in eukaryotic cells, but also the mitochondria, or if you have chloroplasts, they also have DNA within them. Okay, some bacteria can have mitochondria. However, um, the purpose of mitochondria in a eukaryotic cell is that it has that specific DNA which identifies the cell. So if we have a look at the Mark scheme, we've got nucleus. You could have had nucleolus or nucleoli, which is literally inside the nucleus. Um, but you could have also had mitochondria, chloroplast, which contains DNA. Okay, final question for today. It says the resolution of an image obtained using an electron microscope is higher than the resolution of an image obtained using an optical microscope. Explain why. Okay, why is the resolution higher in an electron microscope? Because electrons have a shorter wavelength than light. Okay, if you have a look at the Mark scheme, shorter wavelength between electrons, longer wavelength in light rays. And that would get you 1 mark. Okay, so again, everything that we've already covered in today's lesson.